This is an introduction to lipids in the sea and how we extract them and how we measure them.
Lipids have several important functions, with compounds involved with energy, membranes, and as precursors of bioactive molecules. This is a triglyceride or triacylglycerol, very important energy in the marine environment, that alongside wax esters. And one of the interesting things about marine food webs is the use of lipids for energy storage is much more prevalent in general than it is for carbohydrates. An additional advantage of using lipids is that they provide buoyancy. This is actually a molecular species because I've drawn 3 fatty acids on the glycerol backbone, and they all have 16 carbons and no double bonds, so they're saturated, and that's, in fact, the name is palmitic acid. I drew the same here on this phospholipid. This is phosphatidylcholine. However, in the marine world, fatty acids often have double bonds, and here's a classic one here, eicosapentaenoic acid, easy for me to say, EPA is a handy abbreviation, and this one is often found in both phospholipids and triglycerides, and it is a precursor of highly bioactive molecules called eicosanoids. These are active at the nanogram per gram level, and they used to be known as local hormones. Lipids differ from the other major groups of biomolecules, the carbohydrates, proteins, and nucleic acids, in that they are not polymers of relatively small numbers of different building blocks. This is an amino acid, which is a building block for proteins. This is a nucleotide, and you'll see the different nitrogenous bases. This is adenine, this is cytosine, this is guanine, this is thymine, and there's an H and no OH down here, so this is part of DNA. You'll also recognize the repeating unit of this polysaccharide, chitin. Whereas over here, we're just looking at 2 of the many lipid classes, and the structure differs quite dramatically with the head group. So these exist in highly interconnected metabolic pathways leading to various combinations of fatty acyl chains, backbone structure, and polar head groups. And so if we replace this polar head group with another, we would change this from being phosphatidylcholine to something like phosphatidylethynolamine or phosphatidylserine. It might be easier to see how lipids are built up using a block diagram such as this. And here we have divided the lipids into neutral lipids and polar lipids, and the neutral lipids tend to be mainly storage lipids, so for energy, and polar lipids tend to be mainly components of membranes, critical components of, for instance, the bilayer membranes surrounding cells. And in this case, we can see the glycerol backbone, so C3, and there are 3 fatty acids attached to it. And as we move over to the glycerophospholipids, there are just 2 fatty acids attached, and then we have the polar head group containing phosphate. In this case, we're talking about glycolipids, and so there are sugars involved, such as glucose and galactose, and we have this sphingosine backbone that also contains nitrogen. But once again, we do have fatty acids, and so these are what make these lipids, because these are long chain components that contain just carbon and hydrogen, and so very non-polar. However, you can see in terms of polarity that these would be more polar because of the phosphate groups and the fewer fatty acids compared to, say, triacylglycerols. Lipids differ in polarity, and among the major lipid classes, the least polar are the wax esters, followed by the triacylglycerols, and then the sterols. Together, these 3 major classes comprise the operationally defined neutral lipid classes. And once again, wax esters are the least polar of the fatty acid esters. They generally occur on the surfaces of organisms to protect against water loss, but in aquatic animals they are used as metabolic energy reserves. Wax esters are the major neutral lipids of deep water zooplankton and fish, and of calanoid copepods at all depths. However, triacylglycerols are probably the most common type of energy storage in marine organisms. And this particular sterol here, cholesterol, would be the most common sterol found in marine animals. Phospholipids are essential components of membranes, and they are grouped with polar lipids, including glycolipids. Glycolipids contain one or more molecules of a sugar and are found in bacteria, plants, and animals. Glycoglycerolipids of the form shown are found predominantly in the plant kingdom in association with chloroplasts. The variety of polar head groups gives rise to different phospholipids, which make up about 3/4 of the total lipids in membranes. These membranes form barriers for cells and organelles and act as a solvent within which membrane proteins fold and function. Each specialized membrane has a unique composition. This is phosphatidylcholine, which we've seen before, and this is the head group that would be replaced. In this classic fluid mosaic model, this is the head group of the phospholipid here, and you can see a saturated fatty acid straight here, and an unsaturated fatty acid, which is bent here, and interleaved between these phospholipids, you can actually see sterols. And so this would most likely be cholesterol if we're talking about an animal bilayer. There are many different fatty acids in marine organisms, but they all follow a somewhat similar pattern. So here I'm showing 18 carbon fatty acids. They are all unsaturated, and they're actually biochemically connected in plants. And so this one here has 18 carbons and one double bond. And the shorthand nomenclature we use is 18:1 omega 9. So it's the ratio of carbons to double bonds. And the omega 9 denotes where the double bond is located. And it's based on the Greek alphabet, where omega is the end of the Greek alphabet. And so it's the distal carbon from this functional group, which to a chemist is backwards, but as you'll see soon, biochemists use this in a very handy manner. And so we would count 1, 2, 3, 4, 5, 6, 7, 8, 9. So that's the omega 9 position. When we add another double bond, this is now 18:2 omega 6, it's understood that the double bonds are going to be methylene interrupted. There's a CH2 group here. And so in this case, the total number of carbons is 18, and we count from the distal end, 1, 2, 3, 4, 5, 6, and we know that that's the position of the first double bond, and usually in fatty acids, unless we indicate otherwise, there's a CH2 group in between, and so we know the position of the second double bond. Likewise, 18:3 omega 3, 1, 2, 3. methylene interrupted, and we've defined the structure. What's interesting about these particular fatty acids is that they're biochemically related, but only in plants. And so this additional double bond has been inserted on the omega side of the fatty acid, and only plants can do that. Likewise here. And what that means is that we end up having fatty acids that are essential to animals. And here's an interesting one, 18,2 omega-6, because animals can desaturate on this side, and you could end up with this very important essential fatty acid, 20 with 4 omega-6. And so fish, particularly freshwater fish, are able to do this conversion. And so they would use this as a precursor. They can't desaturate on that side, but they can desaturate on this side. So the omega-6 designation holds. One of the things that is interesting in this particular paper is that they showed that pollution in the case of the whitefish actually decreased the ability of the whitefish to do this very, very important process. We have part of the omega-6 fatty acid series, and these are the two fatty acids we were just talking about, 18,2 omega-6 and 20 with 4 omega-6. And you can see how they are structurally related because they have this omega-6 starting point for the double bond, but they have increased in chain length here, and additional double bonds have been added on this side of the molecule. The relationship between 18,2 omega-6 and 20 with 4 omega-6, also known as arachidonic acid, and this is linoleic acid, is a subject of great interest in nutrition research. And it's important for human nutrition, it's important in the marine world, it's important in aquaculture. And something that gets forgotten occasionally is that this series actually starts much lower down and continues on beyond arachidonic acid. And so you can see 16,2 omega-6 has the same front end and it's shorter. And so what's happened here is we've just done a chain elongation, same number of double bonds. When we go from 18,2 omega-6 to 20 with 4 omega-6, we've done both a chain elongation and increased the double bonds. And then we've done it again here, going to 22 with 5 omega-6, also known as docosahexaenoic pentaenoic acid, DPA. These are actually present in green vegetables, so when we think about terrestrial animal nutrition, including ours, and they're also present in microalgae, very important in freshwater and marine environments. And this is the key product in this series. This has very important functions in terms of being a precursor of the eicosanoids that we mentioned before. And the degree to which you can convert one of these to the next, to this one, or perhaps this one back to that one, known as retroconversion, defines how essential a fatty acid is. And so rats and humans are actually able to start here and then move down to here. This is the desired fatty acid. And there are undoubtedly marine organisms that could do the same thing. Here we have the often talked about omega-3 fatty acids. Again, we're starting from carbon 16 and we're going down to 22 carbons. And you can see the omega-3 designation here. And when I drew the next one, I just increased the number of carbons. And then we move down to 20 with 5 omega-3. eicosapentaenoic acid, or EPA. We have 5 double bonds. Then here we've increased the number of carbons by 2, but we have the same number of double bonds. So this is docosapentaenoic acid, or DPA. And then here we have docosahexaenoic acid, or DHA. And there are 6 double bonds. So we've extended the double bond on this side of DPA. And again, this starting point, the 18 carbon starting point, going to the 20 carbon, is of great interest in nutrition, but in nutrition there's also a great interest in DHA as well. And once again, the ability to move through these different carbon numbers and these different levels of unsaturation defines how essential a fatty acid is. Both EPA and DHA are very important in marine organisms and are known as essential fatty acids. They're required for the normal functioning of marine organisms. Here's a simplified pathway of how we get from carbon 18 to, in this case, it's the omega-3 series, so EPA, and then DHA. And we're talking about animals here, so when you have a desaturase, this is the delta-6 desaturase, you keep the omega designation the same because you remove the double bond from the other side of the molecule. And so we've gone from 18,3 to 18,4 by removing 2 hydrogens and creating a double bond. Then to get to 20 with 4 omega-3, we use a fatty acyl elongase, and then 20 with 4 to the all-important EPA, delta-5 desaturase. And then it's quite a complicated way to get to, again, the all-important DHA, 22 with 6 omega-3. And what's interesting about this pathway is that the same enzymes actually work on the omega-6 pathway as well. In this case, we're showing the 2 series, but starting from the saturate 18,0. A delta-9 would take us to 18,1 omega-9, and both plants and animals can do this. But to go from 18,1 omega-9 to 18,2 omega-6, we've now desaturated on the omega side, as well here, 18,3 omega-3. And so only plants and algae can do this step. All organisms can do this step from here to here, and from here to here, using the delta-6 desaturase, and so on down the series, going down to 20 with 4 omega-6 arachidonic acid, or 20 with 5 omega-3 eicosapentaenoic acid, or 22 with 6 omega-3 docosahexaenoic acid. And these are predominantly present in terrestrial organisms. They are present at a certain extent in some algae, and these are the key aquatic fatty acids. And these are the ones that are of great interest in nutrition, because these are the essential fatty acids that are, these 2 are precursors of eicosanoids. This one is actually a precursor of a docosanoid, and so docosanoids and eicosanoids are these bioactive compounds. And in aquaculture, there is great interest in trying not to use fish to feed fish as much as possible, and trying to substitute terrestrial sustainable products in the feeds. And so trying to encourage marine organisms to do this transition. The interesting thing in the marine world is that these fatty acids, especially EPA and DHA, are naturally present. And so the ability for marine organisms to start here or here and move down is actually somewhat limited. And so this is where there's been great research in aquaculture nutrition. There is another way of getting to the all-important ARA, EPA, and DHA that is found in certain organisms. The polyketide synthases, PKS, use a family of multi-enzyme complexes to produce structurally complex organic molecules or secondary metabolites, collectively termed polyketides, and it's found in bacteria, fungi, and some plants. In algae, long-chain PUFA can be produced by two pathways, either the aerobic pathway using specific discrete desaturase enzymes or proteins, or in some cases, including certain thrustochitrid and possibly flagellates, anaerobically via polyketide pathways. And you will also notice here that I've switched nomenclatures on you, and I've been saying omega, but I've been writing N-. And these two nomenclatures are absolutely identical, but it's left over from the days of typewriters when the only place you'd find an omega would be in Greece. And so the convention started of using N- a long time ago and has continued to this day in many journals. Until recently, the comprehensive analysis of lipids generally involves separation into simpler categories according to their chemical nature. Lipids would be quantified as bulk species and/or as individual components. Advances in chromatography and mass spectrometry, permitting quantitation of lipid molecular species, have greatly propelled our ability to study metabolic pathways and networks. Bulk lipids would be equivalent to total lipids, and you would acquire your lipid extract, and you would do a phase separation, and you could take the extract, you could remove the solvent, and you could simply weigh it, so you can get total lipids by gravimetry. You can also get total lipids by summing the individual lipid classes. And so this is actually an iatroscan run of lipid classes going from the most polar phospholipids to the most non-polar hydrocarbons. And you could just take the sum of all those classes. This is something, in fact, we do routinely, and that would be equivalent to total lipids. And so we've grouped together all the molecular species of all the different phospholipids, the various head groups, the various fatty acid combinations into one peak here. And so this is how we measure the total. And by comparison, we have here a molecular species. You can see we've defined the fatty acids here, and there's the head group, once again, phosphatidylcholine. But you could, and it's very commonly done, hydrolyze these two ester bonds and measure the released fatty acids, the free fatty acids. And this kind of analysis, where you do a fatty acid analysis in combination with a lipid class analysis, is something that we do quite routinely. We see the major interconnections amongst the major lipid classes. And so we have two-headed arrows, we have single-headed arrows, and at the center we have the fatty acids, and these go towards, for instance, glycerophospholipids, but glycerophospholipids can also supply free fatty acids. This essential fatty acid is acquired through the diet and then can go into this sequence here, where it can then go to triacylglycerols, glycerophospholipids, or sphingolipids. And otherwise, this dietary fatty acid could be used as a precursor for the all-important eicosanoids. And you can see what happens is you get cyclization here. Acetyl-CoA can also go through to isoprenoids, which become part of sterols, which can then be esterified to fatty acids. Here we see the basic process for lipid extraction and analysis. Our example here is a shark that is having all its key tissues and organs dissected. We would then extract the lipids in solvent, which we would then clean up in a separatory funnel, and we would draw off the oil from the bottom in its solvent. We would then place that in a warming panel here, so each of these vials contains solvent plus lipid, and nitrogen is blown down into the vial to remove the solvent. And we can do this and then measure the mass of lipid that has been extracted. This gives us total lipid content. We can also take that extract and measure lipid classes in an iatroscan, and that is a combination of thin layer chromatography with flame ionization detection. And in this box you can see a tray with various chromatographic rods, and this is the flame ionization detector here. We could also then sum those lipid classes to get total lipid content. We can methylate the extracts to, first of all, liberate the fatty acids and then make those free fatty acids fly on a gas chromatography column, and we can then measure by GC and GC mass spectrometry. Mass spectrometry gives us the identities, and GC, for instance, with a flame ionization detector, would give us the quantities. And so we separate these now volatile fatty acids after methylation on a column to get this chromatogram, and each of these peaks is a fatty acid that has been methylated, and the area under the curves gives us the quantity. And again, we would identify by the retention time when they come out on the column and by mass spectrometry. Here we have such a fatty acid that has been identified. This is 18,1 omega-9, or oleic acid, and here's the kink right here that is provided by the double bond. We saw the kink in the fatty acids and the structure of the bilayer and the phospholipids, and this is actually very important for providing fluidity to membranes. And so this is the carboxylic acid end of the molecule that would be methylated to make the fatty acid volatile for chromatography, and this is the methyl end of the molecule, or the omega end.